Hello, everybody. So I think we're beginning the next session just before the lunch. And we will talk about empirical studies striking risk. So we have been talking a lot about biosafety and biosecurity. And most of us, when we heard these words, we think on policies, regulation, and we forget sometimes that biosafety itself is a science. So in this session, we will present empirical studies that creates the evidence that helps to create new policy on biosafety and biosecurity, but also to boost the compliance to the existing policies and to prioritize between these policies. So today, we have three amazing speakers. Dr. Rocco Casagrande, that is a founder and chair of the board of Gryphon Scientific. Dr. Gregory Koblentz, that is associate professor and director of the Biodefense graduate program at George Mason University and Schar School of Policy and Government. And Dr. Weiwen Zhang, that is the distinguished professor of synthetic biology and biochemical engineering in the Tianjin University of China. So I have a question for the three of them. And my first question will be, so could you tell us what is your research and how your research informs biosafety and biorisk measures? So I think we will begin by Rocco.
Yes, thank you for the question. So I guess before I say what I'm doing, I should probably start with why I'm doing it, because I'm not a biosafety professional. So over the last 20 years, we've conducted many risk assessments looking at the operations of some of the highest containment labs or policy affecting the pathogen research in the United States and globally. And while doing these risk assessments, we found that unlike in other high-risk industries, there's critical data lacking about what causes incidents and accidents and really the efficacy of various factors that mitigate risk. And you need that evidence because without it, it's very difficult to understand what factors in the laboratory or what components, what equipment, what procedures generate risk. It's difficult to understand what truly effectively mitigates risk and how to invest resources in mitigating risk. So, specifically, the panel yesterday talked about how containment environments differ from country to country, and that's very true. While conducting the gain of function risk and benefit assessment at a panel in front of the US National Academy of Sciences, Ron Fouché said that we had overestimated risk. And when I talked to him at the lunch break about why he thought that, he said that, well, one of your risk pathways is someone spilling a virus culture outside of a biosafety cabinet. And he said that's impossible because we use class 3 biosafety cabinets in Erasmus. And I said, well, Ron, that's really good to know, but our work was focused on the United States, and the very same flu labs in the United States working on the exact same pathogens as you do in Erasmus use an open-face class 2 biosafety cabinet, so you can spill out of it. He was not aware of that working on the exact same virus in these two advanced countries can be done with very different equipment. And the question is, who's right? Is it worth it doing work in a glove box, which is really difficult sometimes and has additional expense associated with it? And the fact is, the evidence isn't there to actually make that decision, so you get two different standards for the exact same pathogen. The other reason why you need evidence is because, scientists value evidence. That's their job. They're creating evidence. And when you tell them why they have to do something that's inconvenient, but you say, we think will bolster safety, they want to see why. What's your evidence? Why do you say that? Otherwise, they're very difficult to convince. And so you need this evidence not just to help with risk assessments, which I like, but also to make smart investments and to boost compliance. And so, after complaining about the lack of evidence for about 10, 12 years, decided to try to do something about it. So I decided to try to find funding for research to fill some of these critical data gaps. And in the process, I didn't find any US government agency or NGO that had the mission to fund this biosafety research. But luckily, the Open Philanthropy Project found us and decided to fund 3 critical lines of research. The first of which is on the physical sciences. And here with our collaborators at UC Berkeley, we're studying the physical processes behind laboratory accidents. So I have some slides here just to show you some cool videos. So what we're doing is we're trying to understand what happens when you drop something. This is a T150 flask, which is commonly used in viral culture, and what it looks like when you drop it with a very, very high-speed camera. This camera takes 70,000 frames per second. It costs more than my car. And it can generate this visceral evidence to people working in the lab about why you might need respiratory protection. These accidents, this is a 96 well plate dropping. These accidents look very, very sloppy, and you can see very fine particles coming off them. And with holography, we can measure exactly how much is an aerosol versus a splash, which is important for understanding risk, but also convincing people to wear various types of PPE. Additionally, what you can do is once you understand the physics of it, it helps with risk assessments, right? But also, you can start intervening in that physics to re-engineer for safety. Here we have a slide with two different Petri plates that are exactly identical, but one has the vibrations dampened in it using marine epoxy. The exact same, the surface that's in contact with the liquid is exactly the same, just one is made to vibrate less. The elastic modulus is higher. And so with that simple intervention, you can create labware that is inherently going to generate less splashes and less aerosols. And that's very cool, because these interventions could be like a penny or 2 pennies per disposable item, because you're just changing the type of plastic or the amount of plastic that you're using. So in addition to the physical science work, I'll hand you that, we're also doing human reliability studies, where we're measuring how often humans make mistakes in the lab. And there, we found that in the life sciences, those data were almost completely absent. So to do our risk assessments, we had to get data on human reliability from other industries, like the rate of fine motor accidents. We had to analogize from aviation when someone was dropping a screw when fixing an airplane engine. And we had to analogize that into the laboratory. That's highly unsatisfactory. So we began to measure the rate at which various laboratorians make accidents while pipetting, how often they splash on the blotter paper, for instance, or on themselves. And in that measurement, we not only are able to, for the first time ever, measure how often per 1,000 manipulations people spill on the bench or on themselves, but also we ask them to call out when they think they made a mistake. So we ask them as they're going to say, okay, I think I touched the tip to the edge of the well, or I touched the tip to my glove or the blotter paper, or the tip fell off. And then we can see how much awareness these trained scientists have of the mistakes they made. And we found that over about 20,000 replicates, about scientists that are working understand about half the mistakes that they make, and the other half are occult. They don't have any idea that they're making them. This evidence is interesting, once again, for risk assessment, but also helps reinforce the maxim that all biosafety officers say, which is every time you work with hazardous materials, you must clean up your entire lab area, and every single time, regardless of whether you make accidents. Now we know why. The answer for why is because you only know half the time you're making a mistake. And we have the evidence for that. The other great thing about these experiments is they're done with a variety of collaborators, some from middle and low-income countries. We had collaborators in Tunisia, collaborators in Jordan. You heard Nisreen Mahmoud yesterday talk. She was one of our collaborators. And a collaborator from a very low-resource clinical lab in Brazil. And with these collaborators, we showed that they can contribute just as much to this research on the safety of pathogens that people can in a very well-equipped laboratory and well-resourced lab, because the main resource you need is worker time. Everyone has time for their workers, and because you just need thousands and thousands of worker hours to do these experiments, so they can contribute very well to it. Lastly, the other line of research that we have is gathering innovations and best practices that are in place in laboratories worldwide. So in this case, in all the work that we've done with risk assessments and containment labs, we found almost everywhere we went, there was a unique practice that was created to address some risk that was found in that laboratory. Either there was an incident and they did a root cause analysis and re-engineered things, or during their process of risk assessment, they identified some residual risk they didn't like, and so went on to create a further mitigation. These innovations we found were largely unique to their laboratory. They weren't being in place elsewhere, except they could have value in more places. The only reason why they weren't in place in more locations is people didn't bother to tell anyone. They didn't know they were being innovative. It was just their job. They didn't appreciate the creative thought that they were doing. So we undertook a project to interview about 60 or so thought leaders in biosafety and research in North America and Europe, and then about 60 or so thought leaders from low and middle income countries from about 50 countries. And we identified their practices, evaluated the ones that could have benefit if deployed more widely, and then brought everyone together to evaluate them jointly and disseminate the practices that we thought had value. And we're slowly writing that up into a book that we'll give out to disseminate that a little bit further. And I think those are the research lines I'd like to talk about today. Greg?
So, Greg?
Great. Thank you. And to understand tomorrow's pathogen research agenda, which is the focus of this conference, you need to understand what the pathogen research enterprise looks like today, and what are the measures in place to ensure that research is being done safely, securely, and responsibly. And that's the primary purpose of the Global Biolabs initiative that I co-direct with Philippe Lensos from King's College London. When we started this initiative back in 2021, our goal was to provide as comprehensive and accurate a picture of the maximum containment labs around the world as possible, given the heightened interest in these labs in the aftermath of COVID-19. We've since went live with the website in May of 2021. Since that time, we've had 90,000 unique visitors. We average about 1,800 views a month to the website, which was rendered beautifully by the talented team at the Boulder Genomic Scientists. And so it provides a very user-friendly interface for both the public and policymakers and researchers to explore the world of high containment laboratories and biorisk management. We've also expanded the site to include not just the BSL-4 labs, but also BSL-3 enhanced labs. In addition, details about national biorisk management policies, including biosafety, biosecurity, and dual-use research oversight. You can find our latest report that we released last month here at the conference, at the table outside, as well as it being available for download from the website at globalbiolabs.org. I unfortunately don't have time to give you the full rundown of the report, so I'll just give you a couple of highlights, and I encourage you to visit the site to get more information if you're interested. So overall, there are more than 100 high-consequence biomedical research facilities around the world conducting research with dangerous pathogens of a variety of sorts. 69 of these labs are what are called BSL-4 labs in the US designation. Other countries have other designations. These 69 labs are either in operation, under construction, or planned in 27 different countries. In addition, there are 57 BSL-3 enhanced laboratories that are BSL-3 labs that have undertaken additional biosafety measures to conduct research that is higher risk than that usually conducted in a BSL-3 laboratory. We've also collected data on the characteristics of these laboratories. So, for example, about 75% of the BSL-4 labs, 80% of the BSL-3 enhanced labs are located in urban areas. In addition, we've tracked the growth of the BSL-4 lab enterprise over the years. And based on our data, about 3/4 of all BSL-4 labs have become operational since 2000. Right? And this was due to a large building boom in high maximum containment laboratories after the anthrax litter attacks in 2001, after SARS in 2003. And in addition, there's been an additional building boom due to the pandemic. We've had 9 countries announce plans to build 12 new BSL-4 labs since the start of the pandemic. For 5 of these countries, this will be their first BSL-4 lab, and most of these new labs will be built in Asia. And I think that helps highlight the areas that need further attention from a biorisk management perspective. We've also, in the latest edition of our report and our website, we've included information on the national scorecards for biorisk management that evaluate the biosafety, biosecurity, and dual-use research oversight policies in place in the different countries that have BSL-4 labs. And this is original data generated by the Global Biolabs team. Speaking of which, we have 2 members of the team in the audience, Myra Emineros from King's College London and Becca Earnhardt from George Mason University. Without them and the rest of the team, this research would not have been possible. Again, just to give you kind of the quick snapshot of where we are at with biorisk management in these countries, biosafety governance is the most well-developed of these 3 areas. 21 countries that we evaluated out of 27 scored high. The weakest areas were in the areas of inventory requirements and required use of PPE. On the biosecurity side, I think 12 countries scored high, and the rest scored medium and low. And in that biosecurity domain, the biggest weakness was the lack of rules about screening DNA synthesis orders for biosecurity. In addition, the number of countries with cybersecurity requirements was also on the low end. The area which we are weakest globally is on dual-use research oversight. Only 1 country, Canada, scored high in that regard. 2 other countries scored medium, and the rest of the 24 countries we studied scored low. Many of those countries received a score of 0, meaning they have no mandatory or voluntary measures to monitor dual-use research in their country. So as you can see, there is still a lot of work to be done in this area to enhance biorisk management around the world. We have a set of recommendations in a report, which I won't go through, but I definitely encourage you to download the report and visit the website. And if you have feedback or comments on our data or on our report, you can contact us via the website, and we're always looking for comments and ways to improve our research. And I'm happy to answer questions when we get to the Q&A session. Thank you.
Okay.
Dr. Weiwen.
Okay, thank you. And I think before I start, I would like to introduce my background a little bit so you can understand what we are thinking. I'm a microbiology by training and also currently a synthetic biology scientist. But unlike the expert in this room, we are not working with pathogen, and we basically use the synthetic biology working with the good bacteria, basically like cyanobacteria, and to use CO2 and generate good compounds. But we are a group, the first group of scientists in China who realized the potential dual-use issue of the synthetic technology, synthetic biology technology, and the related technology. So about 9 years ago, and we start those kind of discussion and what kind of regulation, what kind of mechanism we need to safeguard the progress of the synthetic biology research in China. So we have, since then, we have been doing sort of half time for me and still working on the wet lab, but also working with the policy study. Before I give you some of the example that what we have done in the last 7-8 years, I will give you a couple of figures as, so you know the big picture. So in terms of the synthetic biology research, I think starting in China in 2011, I was among the first grantee receiving the National High-Tech Grant. At that time, the first 5 years is 2011 to 2015. The total money investment in the synthetic biology in China is close to $80 million, $80 million. But in the second 5 years cycle, that's starting from 2018 to the end of last year, 2022, the number increased to $382 million. Under the new circle, a 5-year circle starting from this year, we expect there will be $6 million spent on the synthetic biology. So you can see the number increase significantly, and also the personnel involved in synthetic biology research. For example, I remember that 2011 probably will be less than the people in this room, I think, working on the synthetic biology. But for right now, I just simply calculate the grant number, I think probably close to 10,000 people working on the synthetic biology in China. And give you an example, for example, my university, Tianjin University, is one of the leading synthetic biology research institute in China. We have about 1,000 graduate students working on synthetic biology and 200 or 300 new students coming every year. So that's the scale and also the number we are looking at. So we realized that, I mean, with the fast moving of the synthetic biology technology itself, and also the significant investment from the government, also in recent years, also from the private funding like venture capital and all that. So we need a mechanism to make sure that the technology is really doing good, doing benefit to the human society rather than something going wrong. For example, after the pandemic, when you introduce yourself as a synthetic biology, and the first question people will ask, are you working with viruses? Something like that. I think, I mean, we need a mechanism to doing that. So in the last 7 years, I think we have been making effort in 3 aspects. I will introduce them. And the first one is at the national regulation and the law level. And in 2018, I'm leading the effort, basically writing the drafting the first biosafety protocol related to the new technology, and which was implemented by the Ministry of Science and Technology of China in the summer of that year. In that protocol, basically, the first time we introduced the idea that biosafety is not just related to the pathogen, but also related to the technology itself. So we need to have certain regulation on that. For example, we basically also suggest 3 levels of the regulation for different risk level, basically high risk, low and middle and low risk. And also we suggest that every institution need to have a biosafety committee related to the new technology. And also, it just give you a number. We pull out the number related to synthetic biology out of NSF of China to see how many institutes getting grant related to synthetic biology. The number is 1,200. So you can imagine that a lot of people are doing this work, but without any biosafety or biosecurity regulation on that. So we started to work on that at the one I just mentioned, 2018, that's one example. And also later in 2020, that's the Chinese biosecurity law, actually for the first time, also including the governors of dual-use technology into that law. That's also part of our contribution. So that's we try to set up a high-level regulation at the national level. Meanwhile, we also sort of expand our efforts, also drawing the collaboration from outside. My center is collaborating with Johns Hopkins, and we have a dialogue, track 2 dialogue between the US and China. That's happened 2019 in DC. And we're basically talking about the 2 major countries, what we can do to safeguard the synthetic biology research, which, I mean, the meeting was very good, and unfortunately, it was interrupted by the pandemic. We still hope that we can resume it later the time in the near future. More recently, I actually working within the framework of BWC and collaborating with Johns Hopkins and also the AIP. We proposed a code of conduct called the Tianjin Biosecurity Guideline. Some of you may heard about that. We basically want to build or promote a culture of responsible research, especially in the bioresearch area. So that's the first aspect we have been working. The second aspect is education. Especially after the pandemic, we realized that how much we need regarding the education in terms of the biosafety and the biosecurity. So actually starting around the peak of the pandemic, that's, I remember, it's March of the 2020 in China, that's pretty much the peak time. And we started several courses open to the general student. Actually, 85% of students coming from different, from the major outside of biology, like a computer, social study, and the law school, and so on. And so far, we have put through more than 1,000 students out of the training at the end of last year, so within 2 years. So the idea is, in the future, I guess, by economy or by industry, basically, will utilize expertise from all major, and we don't want to limit our education to the bio student. We want to have education, more general biosafety education for general students. So for that kind of work, we also realized that how limited for the textbook and the teaching protocol and all that. So we are actually the first university basically implement those kind of education for engineer school. And so most of them probably will also go into, either they go into the government job or go into the industrial job. They probably will also relate to the biology or biosafety. So we give training to them and make some good effort. And more recently, we working with our colleague in UK, and we are writing a first textbook on biosecurity and biosafety. I hope it can be finished this year. The last aspect we have been working is tools. And I think both computational tools and the experiment tools for synthetic biology. For example, one of the work we finished recently is through the process of synthetic biology research, we try to identify the risk on each of the steps. And then we send out a questionnaire to the PI of the national project to ask them to identify the most possible risk step. And of course, there are many of them. And it's just turned out that the attitude towards biosafety turned out to be the top one, and the people think is the most important fact related to the safety and the biosafety of the lab. Other facts like infrastructure, quality, and years experience, and also the lab hygiene condition, and all that actually come lower than that. That's very interesting finding. We found that, and also highlight the importance of the biosecurity and the biosafety education in China and also in other countries as well. I think that's pretty much the three aspects we have been doing, and also a little bit introduction to our institution. Thank you.
Thank you. So I have a second question before going to the public, and that would be like, what were the main challenges you faced during your research, and what lessons you learned from these challenges? So we can begin again by Rocco.
Sure. Well, the first challenge was finding an organization to fund the work, which I must say is still a challenge. I'm kind of surprised that we're several years later and there still isn't really an organization within the US that understands biosafety research as their primary mission. The second is we were working with researchers who either had not thought about biosafety before. So the data I showed you on the splashes is from people who are in the mechanical engineering department at Berkeley, but they had the tools to investigate some of these phenomena that we knew informed biosafety. So convincing them that these are interesting problems and that they should pivot their lab to this work was at first difficult, but since that time they realize how many data gaps there are and how fruitful this field of research can be, so they want to continue on. The secondly was engaging people who were already in the field of biosafety, but not on the research side. So our collaborators at the University of Maryland and Colorado State are in environmental health and safety. It was difficult for us even to give them money and for them to accept it to do work because they're in the overhead side of the university, not in the research side. So that was also a difficulty.
So what about you, Greg?
So we had a number of challenges. I mean, one of them relates to the fact that there's the terminology biosafety, biosecurity, dual-use research is not always used consistently, both between countries and even within countries. The way that countries designate biosafety levels and containment levels varies, particularly when it comes to the BSL-3 enhanced labs. There's very little national guidance on what that means. There's no international guidance whatsoever, and so there's a lot of variability in what labs consider to be enough additional safety measures to consider themselves a BSL-3 enhanced lab. A lot of these terms get used interchangeably. We studied 27 different countries, so there were language barriers to making sure that we're finding the right documents, because just the the absence of the document doesn't mean they don't have regulations. Maybe we just can't find it. So that was a very kind of long process to make sure we were actually gathering all the right data and we were getting it translated properly. And then I think building the map was a challenge in terms of making sure that we had the right locations for laboratories and affiliations, institutions, and that sort of thing. And again, we took a lot of care to make sure we were getting it right. But that is a whole separate skill set to do kind of the geospatial analysis and do the geolocation. So I think we did a good job overcoming those challenges. But again, if people do find any ways to improve the site, we definitely would welcome the feedback.
Okay. So, and for Weiwen, I think the same question, but I will have even in this field of synthetic biology, there's the research itself, but it has been also a movement of open research. Yes. So how do they interact, or how do they see when there's guidelines or there's regulation that trying to be included in these fields by other organizations or by governments that are not part of that movement? So, yeah.
Okay. I will try to answer the first part of the question first. That's the challenge. I think I can see, in the last several years of working in this particular area, I think I can see two major challenges we have been facing. The first one is actually the attitude from scientists. I think, myself is a scientist, and we always think that as long as my intention is good, I have every right to do everything I want. So that's what science should be, right? I guess, that's somehow when we start to talk to people and discuss with people the potential issue, and I don't think everybody agree with us at the very beginning. I think most of us, I somehow recalled that I was giving a talk in Tsinghua University. I think that's before the pandemic, and several of the people in the audience started to question the potential. How much, how much the probability on that kind of things actually happen, and whether we need those kind of regulation for that. So that's one of the challenge, I think, through the pandemic, I think people are changing. Somehow, at least, I can see that kind of trends is changing. The second one is we are still working on that. That's sort of like the balance between the free science and the innovation and also the governance. I don't have a very good answer to that yet, but also that's different in different countries, basically. For example, in China, we really hope that bioeconomy can be part of the overall GDP, maybe a higher percentage and all that. But at the same time, almost everyone, especially from industry, they don't like this kind of governance or regulation because that means cost. So we also facing a lot of those kind of issue, and up to now, even we have a China biosecurity law basically cover the dual use issue. But when we go in further for the real practical regulation, still have a long way. Basically just cannot get a good balance or cannot reach agreement what would be the best balance on that. Okay, that's the two major challenge. And to your second part of the question, I think for example, for the DIY group, and even the industry, industry, of course, they have different size. Some big company, they probably capable of doing, accept certain amount of regulation. They have people to deal with that. But when you think about the smaller one, like startup, for example, in 2018, when we proposed that every institution need to have a biosafety committee, and immediately the staff said, I'm not capable, and I cannot afford on it. So those are the real issues that we don't have a very good answer yet. But alternatively, we are thinking about, okay, if initially we are thinking about on the national level, and then the second level will be at the institution level. If not happening, then whether we have sort of intermediate, then we have another level, basically draw the expertise from the nearby university and institution to service to the industry and all that. So So those are the issues I think a little bit related to your question. But regarding the people are doing the work in the garage, I don't think right now actually we have a good answer to that. I don't think they even know what the regulation is look like in the country. A lot of them just treat science as a free hobby. I can do whatever I want as long as my intention is good. It's actually has been discussed in multiple location and the meeting. I don't think I have a good one answer yet, but I welcome any comments on that. Yeah, thanks.
Thank you, thank you. So I think we're just in time for questions from the public about, yeah, so we have 3 questions, that's perfect.
Jonas Handbrink of Oxford University. Thank you very much for all the really interesting results, and I think this is exactly what we need to make better policies. I want to briefly plug some of my own work here. We're currently putting together a bibliometric analysis of enhancement of potential pandemic pathogen studies in influenza virus research. And we have some very preliminary results. I think overall we see that we identified about 50 studies over the last 12 years. And there was an increase in the last year. In 2022, we found a lot more studies. And overall, over the years, we also saw that there was a shift towards an increasing fraction of the research coming from Asia. And overall, I just want to say, if someone has any input, I'm very keen to make this study as helpful and robust and non-polarized as possible. So if anyone wants to talk to me about that, please do. One more question. Jens was saying about the BSL-3, BSL-4 lab challenge, that actually the more potential pandemic pathogens are being studied in the BSL-3. Greg, how do you see that impacting on the global biolabs work?
Yeah, so...
One member's supposed to take 3 questions.
Oh, yeah.
3 questions.
It's tempting, though.
Thank you all. I just want to thank you guys for your talks, but I would argue that biosafety as a profession is falling behind in synthetic biology worldwide and in silico. So I guess I have a question about what your thoughts are on how to improve it, and whether you'd make your guidelines more accessible to others around the world, for example. But kind of, and maybe to Rocco, I just want to make a comment. I don't know about funding, but during the pandemic, we were constantly dropping tubes of saliva when we were doing our COVID testing, and we didn't have any good data to say how far you could, have your next person spitting into a tube, because we did saliva testing for COVID, and we dropped them all the time. So I'm sure we caused a few people to get sick. But then, how do you build trust? Just kind of getting to the last panel, this is my question. How do you build trust based on the work that you're doing and not instill fear based on watching those things drop? How do you make the public, or not how do you make the public, I don't want to manipulate the public. How do you argue that the work that you're doing is actually for good, and how do you build trust?
Yeah, that's our approach.
So this is for Rocco. Thanks very much for the work you're doing. You and I and others have had lots of conversations about the lack of empirical evidence behind biosafety. And I think it's very important that someone should be taking the lead in this, so thanks for that. Has anyone started looking at how organizational structures for considering risk and managing risk impact the efficacy of the response?
Okay, so I think we can begin. Maybe you can go first with the BSL-3 question, Greg?
Sure. So we started with the BSL-4 labs in 2021, in part because they had the highest profile in the public due to the pandemic, and also, quite frankly, we knew there was a limited number of them, and so it was a way to start the process and get information out there. But we recognized that these were not the only labs that were conducting potentially risky research, and we said so at the time as well. When we moved to the next phase, we did think about looking at all BSL-3s, but I know from other studies that have been done that there are probably thousands of these labs around the world, and that was a little bit daunting. So we decided to expand our scope to the BSL-3 enhanced labs, and those are the labs that have actually been used, for example, to work with reconstructed 1918 virus, to do the gain-of-function research with mammal transmissible H5N1. So those are the labs that typically have been used to conduct research that is of concern for producing enhanced potential pandemic pathogens. Does not mean that there is not similar work being done at BSL-3 level or even lower, but given that these labs are promoted and described for that purpose, that's why we focused on them. If someone has the funding for us to go out and count the thousands of BSL-3 labs, we'd be happy to do that. But we recognize that would be a major undertaking. So we're trying to keep the scope limited, but acknowledging that there is other activity, other research, other facilities of concern. But it's a lot harder to map those than the ones we've identified.
I will comment on the synthetic biology question. It's, I guess, especially not just synthetic biology. For most of the sort of cutting-edge new technology research area, it seems to me it's almost like the quickest, the better. It's basically you try to come up with a new idea, and so that's how the society move forward. What we are trying to do is basically just to sort of ask people basically, or implement a checkpoint. Just think about the biosecurity and the biosafety consequence related to this research. I don't think anybody, or most of the people, when they do the cutting-edge research, they take that into consideration. I think they just do it, and I'm small scale, I'll just do it. But at this moment, even the small scale, I think, it can be going very fast. I think with new technology, for example, the AI-based design and also the high throughput technology, even a small idea can actually have a very big impact later on. I recall when the COVID-19 DNA sequence was released. I just recall that George said that's January of 2020, and then somebody in the community already started to synthesize the spike protein, the gene, and the synthesis of the gene and put them in the vector, then ask everybody, if you want to do research, I will share with you. I was a little bit shocked at that moment. I think that's February of 2020, because the gene encoding the spike protein has been synthesized and freely transfer between people, and there's no regulation on that. Of course, later on, since the thing is too big, nobody cares about the small thing. But just think about the risk. If you have the similar things happened, not at this scale, at COVID-19, and the scientists just freely synthesize the whatever gene they want and give to people to doing research, that's actually a very significant risk associated to that. So that's why we are thinking sort of build a culture that for the scientists, when you start something new, and just have a second thought or give a checkpoint over there to think about any association or any issue with the biosafety and the biosecurity. But overall, I think, that's one end at the sort of scientist level. That's why we promote the code of conduct for scientists. But at the same time, I think that's probably not enough. I think that's why different level of regulation need to be kicked in. I think that's what I see for the issue. Thanks.
Thank you.
And then to Tim's question regarding the role of organizational and administrative setup in biosafety, I'm not aware of any other studies that are going on that way that are very evidence-driven. I have to say, there's a lot of really interesting studies that could be done. So for one, in the United States, we have some institutions that centralize shipping and receiving of pathogens, and others let it up to the lab level. And you get very interesting differences in the types of incidents that occur, whether you have professionals packing and receiving pathogen shipments versus not. and there's reasons for them to be set up in the different ways. So just understanding what the costs and benefits are of those different setups, and then what the effect is on safety and incidents would be extremely interesting. One of the things that stymied that type of research is an inability to measure anything, because luckily accidents are very, very rare. So you don't really have a lot of opportunity to measure. So one of the studies we just did was in collaboration with the University of Chicago and George Mason University and Texas AMU. They were able to share with us their records of violations of laboratories upon inspections. And we were able to look at all the different laboratories run by various PIs and look at their rate of violations over the years, and importantly, the rate where they got cited for the exact same violation multiple years running. And we found that with this very objective and transparent and evidence-based approach, we found that indeed there are just a handful of labs, roughly 20% of labs, that are responsible for 80% of the repeat violations in an institution. So there's something about this small minority of labs that isn't embracing the culture of biosafety. And now that we can measure it in a way that it's not just the biosafety officer doesn't like me, right? Well, now that we can measure it in a transparent way, there's a lot that you can do to start understanding, well, what are the differences between these low-performing and the vast majority of labs? What is it about the way the PI interacts with the lab? What is it about senior postdocs in the lab? What is it about their operations? So you can begin to answer those types of questions. To answer David's question about how these types of studies don't kind of reinforce the fear, I would say there's two things there. One is our studies that we publish, we always say that we're doing this because there's an intervention, right? We understand the rate at which Eppendorf tubes splash when you open them to determine whether you should use Eppendorf tubes versus screw cap tubes, versus open them in a biosafety cabinet more frequently, versus clean up more frequently. There's always a mitigation. You just have to know what the costs and benefits of that mitigation are, so you need that research. Secondly, and this was kind of epitomized during the gain of function risk and benefit debate, there's often a disconnect in the public discourse between an incident and a pandemic. I drop the T150 flask and all of a sudden there's a global pandemic. When in fact, there's many, many levels of causation between those, all of which you can interrupt. So you drop the flask, then you fail to get out of the way, you fail to self-decontaminate, your PPE is also faulty for some reason, and then you get a lab-acquired infection. And then that person fails their occupational health and safety protocol, so they don't necessarily isolate, or they don't do whatever, and then they interact with the public, and that causes a secondary transmission. And then there's also a failure of nature in that the virus doesn't self-extinguish. There's all these cascading failures that have to occur between oops and pandemic that a lot of people don't get, and we try to always communicate that as well. I think lastly, related to Jonas, what you said, and also what Jens was saying earlier, related to the levels of biosafety and how biosafety tends to always be the science of go slower or the science of more, do more, because the reason why it's that way is we don't really have evidence. All we can say is, it seems kind of risky, let's also put this extra engineering control or this extra environmental control. And what I hope the biggest impact of this research is we understand the actual efficacy, the cost efficacy of each one of these things we're putting in place, which makes biosafety more sustainable. You do only the practices and buy only the equipment that is actually buying down risk. Everything else goes to support the scientific enterprise. And that's not only good for science in like the Western world, but also when you think about the global South. Right now, it's really hurting sustainability because we outfit these labs to Western standards when we might not need to. They could potentially do the work just as safely with half the equipment that they already struggle to maintain. So these data hopefully will help boost the practice of science worldwide by doing exactly what you need to do and no more.
And if I just build on that, I think there's particularly significant gap for biosafety research on the BSL-3 enhanced level, because there's a kind of a menu of options that labs can put in place to be enhanced, and yet I'm not aware of any research that's been done that correlates these additional safety precautions with actual reduced risk posed by whatever experiments or pathogens they're working with. So I think that is a big hole in the biosafety kind of research literature.
Mm-hmm.
I think maybe one or two more questions because there's no more time in the clock. So, oh my God, there's many.
5 questions. 10 minutes.
Okay, nice, thank you.
Okay.
Hi, this is a question for Greg. So very interested to see the information that you all put together. I've been very impressed. And I think it kind of ties into some of the discussions we've had in earlier panels about the issue of transparency. But not all individuals, institutions, governments have same perspectives on transparency or the exact value of it or what type of information we should be transparent about. So I'm just kind of curious in terms of whether your work has generated sort of negative reactions. You don't have to name names or anything like that, but just if it has, and if it hasn't, that's great. That's a good sign. But if it has, I'm just kind of curious about what kind of resistance you might be receiving in terms of making this information out there and available. Thank you.
Hi, quick question for Rocco. We know that when we talked a lot about risk assessment, but there are two sides to that, or two elements to that. The risk side, which is what we focused on here, but there's also the benefit side. And I know you've done a lot of work actually on the benefits side and how we elaborate that. And I was just wondering if you could quickly speak to that, just to put that on our map as well. Thanks.
Yeah, it is still 2 more.
Good morning, Johnny Nemi from WHO. I would like really to thank this panel. It was really insightful to see the evidence-based approach. I really commend this evidence-based approach, and I really think that scientists, we should be focusing all our risk information and all our approach to this evidence-based. And I'm very sorry to hear that there is a lack of funding for risk evidence, because in my opinion, this is a bottleneck that could really solve many of the problems that were discussed in the previous panels. The lack of trust, lack of discussion between governance and science, etc. I think our role as scientists is to be solid in evidence-based science and not talk about policy. It's not our job. We just stand behind our scientific results. And if I want to take one thing after this panel, is that make all the best to enhance and support this evidence-based approach and a risk management approach to biosafety and biosecurity, because all these issues you mentioned are really insightful, and I would really hope that a fora like this one would encourage donors to support this kind of approach. Thank you very much.
I think there was one last one.
Yes, hello. So I have a question about international cooperation. So the cooperation has been damaged, as has been said by Dr. Gao earlier. So, like, what are like the opportunities to restart, relaunch those cooperation, especially in the very tense geopolitical context between China and the US?
Okay, so I think we can begin maybe by Greg and the reaction to the BSL-3 studies.
Sure. So transparency was a very important part of our project from the beginning, and all of the information we used was from open sources, from the websites of these laboratories, scientific articles, the public CBMs that countries submitted under the Biological Weapons Convention, because they are required to declare any BSL-4 labs that they have. So all of that data was incredibly useful. And then we used that then to contact the labs and confirm the information that we were putting on the website. So our goal was to have this transparency, in part to provide this information to the public and policymakers and researchers, but also kind of to help demystify what these labs are and what they do, and not make them seem mysterious and and dangerous in any way. And I think some of the research we did has kind of dispelled some of the myths or misperceptions that you have in the public. So, for example, there is a notion that all the BSL-4 labs are all these gigantic research laboratories with thousands of square feet of lab space. The reality is, half of the BSL-4 labs are less than 200 square meters in size. Right? That's the size of a tennis court. Only, I think, less than 10 of these labs actually are more than 1,000 square meters in size. So they're just not the ones that are typically seen in the media. In addition, there's, I think, a misperception that most of these labs are military labs or top secret labs. The reality is most of them are public health institutions that are run by governments that are engaged in public health missions. So I think by providing this information, we're able to help kind of demystify what these labs do and how they operate. And I think that's to the value for the BSL-4 community, and especially in light of we are living now in an age of disinformation, and there's a lot of bad information out there about BSL-4 labs in general. I think we're trying to counteract that and provide an antidote by being as open and transparent as we can, and providing as much complete and accurate information as we can about these facilities.
So, Greg, to follow this question about the reaction of the countries where you were studying, so what happened with the countries where you ranked that the guidelines were around 0? So how do they react to that? And if there was a guideline or help that we can give to these countries to try to upgrade quickly the ranking, because they already have the labs, so...
Yeah, so far I don't think we've encountered any resistance to our findings or disputing our findings. And in terms of what can be done, again, we have a whole bunch of kind of recommendations in the report. One thing I also highlight is the global guidance framework for responsible use of life sciences that the WHO put out last year has, again, a number of recommendations that any country can put into place. As well as things like ISO 35001, which is the international standard for laboratory biosafety and biosecurity, which provides a ready-made template that any lab can adapt. And also, it's worth noting that among the countries that tended to score lowest on biorisk management, including biosafety, biosecurity, and duty research oversight, many of those countries don't yet have operational BSL-4 labs. They're the ones that are planning to have these labs, or they're under construction. So there is still time to help those countries enhance their biorisk management policies and make sure they have the right legal regulatory infrastructure in place, so that when the laboratory infrastructure is operational, they'll have the right governance measures in place as well. So again, I think one of the values of this research has been to show where are our current weak points and how much time do we have to help put in place some interventions. And I think especially the panel yesterday that had the International Federation of Biosafety Association on it, and the International Experts Group of Biosafety and Biosecurity Regulators, those are the kinds of organizations that can provide the assistance to help make sure those countries can enhance their biorisk management policies in time for them to be operating their labs safely, securely, and responsibly.
Thank you. So I think we can continue with the other questions.
Sure. Philippa, to answer the question about assessing benefits, yeah, if you look at what we did in the risk and benefit assessment for gain of function, we don't expect anyone to be able to replicate that. That was thousands of persons' hours of research, understanding what the scientists themselves thought were the benefits, and then talking to pharmaceutical companies, people doing biosurveillance, governments, et cetera, about how those data are actually used. And no one can do that. So the benefit assessment, understanding the benefits of pathogen research, has always been the missing leg of the stool when evaluating the value of research. So scientific merit has always been part of the analysis. And then we've had biosafety risks and biosecurity risks. But there was never the piece of, well, what is the benefit to public health and medicine of this research? Because that's been too difficult to do. So a while back, we got funding from the Future Fund under the direction of Jonas. And we developed a very simple scheme that anyone could use. So like a group leader or even some government employee could use to assess benefits of research by understanding the likelihood that the pathogen that's under study will circulate and cause harm, which is a concept that Gustavo mentioned just yesterday. It's that basic concept of how likely is this pathogen to cause harm. And then also how well known or how much anticipated any modification to that pathogen is going to be. Like if you know that there's evidence in nature that there's like genetic shift or genetic drift to evade immunological response that's going to create this pathogen or modify it in the future, then it has a lot more benefits to research that than a change that's just merely hypothetical, something that could occur, but there's no evidence that could occur. So by using those two factors to understand the potential benefits of research, and those are data that you could find basically from Wikipedia, right? It's not something that's hard to find. You can kind of place any pathogen research on a field of how much benefit it would be so that you have the evidence to weigh its scientific merit and biosafety and biosecurity risk to make a better informed decision. And once again, speaking of funding, because the Future Fund no longer exists, that work is kind of on pause.
Thank you.
I will finish.
Yeah, please.
Okay. I will comment on the funding issue and also the international collaboration. I think, especially in the synthetic biology area, I agree that the funding actually for the policy and also for the biosafety research are really limited. I think until last year, we finally convinced the government agency to fund the first project focused on the risk analysis related to the synthetic biology. That's so far only one in China. But meanwhile, we see the needs even for like quantitative methodology and for the fast-moving research area and very dynamic, and you need very sophisticated technology and analyze tool to deal with that. Those are not available right now. I think need funding to do that, so we can answer the question like I mentioned before, how to strike a better balance between the innovation and also the risk management. So I agree with that. In terms of the international collaboration and all that, I think actually I personally saw that, for example, the United Nations organization like BWC and the WHO already taking lead on that. I think I sort of hope that those kind of lead can be more significant. I think, for us, we do have individual collaboration across the country, but those are very limited. I think at the UN level, if this kind of platform, or can provide this kind of a platform for the international collaboration focus on certain area like synthetic biology research and the risk related to that, that would be wonderful. So that's just my comments. Thanks.
Yeah, thank you. So I think the time is up, so we have to wrap up this session. So I think what I really like of this session, as the WHO in speaker talk, is like we really see the necessity of evidence in biosafety. I think it will really help all the different points we have been talking in this meeting. First, to really design measures we can prioritize that are really necessary, and not to have the impression that we are mitigating risk, but maybe it's not the right answer that we're giving. Second, to the researchers, so we can ourselves understand the importance of each of the measures, so we can really understand the compliance, the necessity that we have to do. And third, I think we have talked a lot about the trust. So if we can really explain these are the risks, these are the benefits, and this is all the measures, so we can mitigate the risk to have them almost zero, because risk zero do not really exist, then I think it will be easier to build trust for the public as well as for the regulators. And on the international cooperation, I think sadly we don't have time. I think there's a lot of different issues we have to see, because regulations are so different between countries, and because we do not all have the same capacities, there's a lot of time and unequal exchange in collaboration, especially with the high-income countries to the developing countries. There's a lot of things they ask us that they know we cannot complain, but then at the same time, I think some exchanges, even of material, is a lot easier for us to send our viruses to them, and then we cannot even have them back to work on them. So this is another issue that we have to talk about, regulation between exchange of materials and as well as other intellectual properties, biosafety, and how to help interaction and collaboration between countries so all of us can work in pathogens that will be of global importance. And thank you very much. I hope you enjoyed this session.